This is an owl. He has normal vision and he can perceive all the usual colors owls can see. This is an owl with color blindness. Some of his genes were mutated, which caused his mutation of color blindness. The owl without the vision deficiency is able to see all of the colors on the visible spectrum, ranging from the short wavelengths of the violet side to the longer wavelengths of the red side. On the other hand, this owl can only see certain amounts of colors because of its red-green color blindness. Why can I see less than him? What happened to me? What happened is that two of these owls' genes were affected, causing it to not perceive the long and medium wavelength colors to the fullest potential. In this case, the OPN1LW and OPN1MW genes were affected. These genes are responsible for constructing proteins so that the organism can see colors on the green to red side of the visible light spectrum. Now that the genes are affected, the correct protein is not fully constructed to give the ability of saturated color vision to the owl. There are 3 opsin proteins that are made for the purpose of perceiving light, and those include one for OPN1LW, OPN1MW, and OPN1SW, which is the short wavelength side of the visible spectrum. In this scenario of the owl, the 2 opsin proteins are made abnormally in the OPN1LW and OPN1MW genes, meaning that the owl cannot see red to green colors very easily and in many shades. Opsin proteins are light sensitive, and when they are created normally, they will connect this molecule called retinol. This chemical bonds to light-sensitive proteins such as the opsins animals need for seeing color correctly. But that's so unfair. Why do I have to deal with this downside? That's because when they bind together, light strikes the opsin and a signal is sent to the usage of retinol to send a signal to the brain to receive color. Opsin is light sensitive, so when a long wavelength of light such as red hits it, a signal will be sent to the brain to receive and depict a red color. This does not function correctly when the opsin protein for the long wavelength gene is mutated. This means that light that is meant to be red can be perceived as a much lower value of red, or maybe not even red at all. Whoa, look at that apple hanging on the tree over there. It looks so good. What apple? Where is it? All I see is barely any green or red when I look at that tree. Maybe your genes are mutated and you don't make the necessary opsin proteins to be able to see colors correctly. Since you can't see the red apple and the green tree, maybe you have red-green color blindness. That means that you aren't able to see high amounts of green and red shades. Wow, I never knew that. It's really annoying that I can't see many of those colors. I never knew that my vision was different from yours and everyone else's. Anyone's DNA can be mutated, and you were just one of them who caught the chance of your vision for your cone cells being different. It sounds like your OPN1LW and OPN1MW genes were mutated. Those are the medium and long wavelength light perceiving genes. When you make opsin proteins to be able to see, they likely don't function as they should, or they are a different structure than what you want them to be. These proteins interact to pick up visible light and transfer it to the brain through the use of a chemical called retinol. When these 2 chemical structures work hand in hand, they send a signal to your brain to detect a certain wavelength of light to transfer it into color. In your case, the opsin proteins don't work as they should, and you have a minor disability of not being able to see the color on the fullest scale. When you look at that apple, since your genes are mutated, your opsin proteins don't function as they should be able to, so you don't see the full red color of it. Instead, you see a faded out shade of red or hardly any red at all. With your mutation, it probably means that one of your parents most likely had a color blindness mutation, and that's why you also have one. That's really not good. I wish I was just normal like everyone else and be able to see everything I should. Well, it's not that bad. It means you are unique and it shouldn't upset you too much. You just have a slight variation in vision compared to most other owls, but there are still owls out there that have similar color blindness issues just like you. We need to split this DNA and add an RNA molecule to it. I'm going to split the DNA in half between the 2 strands and break all the hydrogen bonds connecting the bases of each strand together. We're going to need to produce the complementary mRNA strand to the template DNA strand. We're going to need thousands of bases to add complementary to this DNA strand for both the OPN1LW and OPN1MW genes. We can't make a mistake as we go along through to add the bases. If we make a mistake, it will not end up well, and there may be a mutation calling for color blindness. If we make sure to add the right bases, no mistakes should be made, and this owl will be able to see full color. Since the OPN1 LW gene has a specific base pairing, we will need to add the exact complementary bases to all of these. When adding the complementary bases to the template DNA strand, we will need to make sure to change adenine to uracil instead of thymine, which would be for complementary DNA. Since this is mRNA, the strand will be slightly different when the bases are added. DNA is different from RNA because DNA is double-stranded and RNA is single-stranded. This is a picture of DNA. As you can see, the sugar of the DNA does not have an oxygen on the 2' compared to the RNA ribose with oxygen. And this is a full picture of an RNA strand. And as you can see, it is single-stranded. For adenine, we need to add uracil. For cytosine, we need to add guanine. It is like a normal process, but we don't want to make any mistakes or else the owl might be not able to perceive regular color if we mess up somewhere on the strand. Every base pair should be exact and correct to ensure no mistakes will be made further on. Make sure that everyone is adding the right bases, and we need emphasis on that. mRNA is being brought over to the ribosome after it finishes being transcribed. Now it is ready to be translated inside the ribosome to make the correct amino acid sequence for the OPN1 LW and OPN1 MW genes. Anticodons from the tRNA strand are placed along with the codons on the mRNA to create specific necessary amino acids. Proteins, also known as polypeptides, are produced from this process after all the amino acids have been formed to bond together with peptide bonds. Translation is the next process necessary to make the protein chain connected by peptide bonds. There are various amino acids used in the formation of proteins for all types of genes. In color blindness, this is the step where the protein of opsin should be produced. Oh, this is when my opsin proteins are supposed to be made, but they aren't. In a regular case of vision, opsins are made for all genes of color perception, including the OPN1 SW, MW, and LW genes. Each gene makes an opsin to become detected by the brain as a certain wavelength of light, translating it into a color perceived by the organism. In the owl scenario, opsin is not made correctly in the OPN1 MW and OPN1 LW genes, meaning that the owl cannot see red to green colors very easily, and they are a darker shade than they should be normally. Translation sounds so weird, and I'm still wondering how it generally works and how the wrong protein is made. During the process of translation, mRNA is brought to the ribosome and transported along the smaller ribosomal subunit. It is passed alongside and it represents codons of 3 nitrogenous bases consisting of adenine, uracil, cytosine, and guanine. Each codon codes for an anticodon to be attached to it. The anticodons are complementary to the codon, meaning that the anticodons are complete opposites and interact perfectly with the codons. For example, if a base sequence reads AUG on the codon, the anticodon will read UAC. Anticodons are a 3-base pair sequence ready to pair with a codon to ensure that the correct amino acid in the protein chain will be produced. Normally, the correct anticodon will be brought to the codon to form the correct amino acid, but in the case of color blindness, since the mRNA sequence is affected and the codons are different as they should be, an incorrect anticodon is brought over and affects how opsins are produced. Oh, wait, I understand it now. My mRNA strand is messed up because my DNA and my tRNA brings over the incorrect amino acid for my light-perceiving opsin proteins to be made. The necessary opsin for perceiving red and green color spectrums are muted because the incorrect amino acid sequence is formed after the anticodons are wrong and not complementary to the incorrect and affected codon on the mRNA. With all of this, it means that the owl cannot see red to green colors of light, and the opsin proteins are made incorrectly. I will need the mRNA strand to work with the tRNA to make an amino acid chain so that the opsins are made correctly for the owl to see color as they should. I have all the components to be able to pair with the tRNA. I do too. We can both make the correct amino acid sequence and ensure that the opsin protein is made as it should be. So, during my process of RNA translation, my color blindness genes became mutated? Yeah, your OPN1 LW and OPN1 MW genes were affected, and that explains why you can't see. Your transcribing did not go to plan, and the incorrect codon was formed, so that means that your tRNA strand initially brought over the right amino acid, but it didn't matter because the sequence in the mRNA was altered. This leaves you with the inability to produce the necessary opsin to perceive long and medium wavelengths of light, so you are colorblind. That's so annoying. I never knew all about that until you told me about it. I wish I could see more color and be able to see the world in a more normal way. I wish that my process of transcribing DNA into mRNA just never got messed up, or else I wouldn't have to deal with all this sadness. But you should look on the bright side, because many owls have color blindness too, and at least you didn't get a huge difference in your DNA. Some owls have frameshift mutations which cause massive problems because they delete or add bases to their mRNA sequence. At least you only have a minor mutation, where you have a missense point substitution change. This means that only one of your bases was altered in one of your codons, meaning that the incorrect amino acid was made, but it did not have significant negative effects towards you. With a frameshift mutation, so many of your codons would be messed up, and you would have significant changes in the proteins that you make. I guess that's true. I shouldn't worry too much about it, but it's just sad I will never be able to experience real color like most other owls. But I understand your point, and I'm glad I don't have a significant mutation that changes almost everything about me. I guess I'll have to continue forward with this knowledge about my colorblindness missense point mutation. Thank you so much for informing me about all these things. I feel more knowledgeable now, and I'm looking forward to my future.